He's a bioinformatician by training, especially on programming languages, and more specifically on the optimization of functional languages. So let's hear more about unity through diversity, building a legume federation with the legume information system, and friends from Andrew.
Okay, well, thank you very much again to the organizers for inviting me here, and to you all for being willing to listen to me. I did want to start out by saying that although my talk will be about the legume work that we're doing, I think that the ideas that will be presented here are applicable, to many different species. And I was dismayed to see that Dave Marshall would be talking before me, because I thought, oh my gosh, what a tough act to follow. But I think it will complement what he presented pretty nicely. So I'm grateful to you for setting this up. Okay, so the Legume Federation is an NSF-funded project that a number of groups in the United States have, who all work on legumes in one way or another. So, I've been involved in the legume information system for many years. And I found this little quote from a paper that came out several years ago. And I actually was presenting this before the project had really gotten off the ground at PAG a few years ago. And I worked up a little use case around this paper. And I thought that this description in the paper was a really nice way of kind of saying what we're really all about. Basically, we're trying to coordinate various groups with expertise in different legumes, and basically allow comparison between them to really improve the information content of any of the resources individually. So I worked through some examples using the tools and data resources that were available at the time, which were kind of clumsy. But Tim Close, who was the senior author on this paper, happened to be in the audience watching my presentation, and he came up to me afterwards and, of course, was interested in the fact that I was presenting on his data. But he was also kind of interested in the project because they have been working on a cowpea genome, and he was interested to see, well, what could this new initiative do for us? So I thought I'd start out today, similarly to David, by presenting a use case, just kind of showing where we've gotten to with the project so far. And this is going to be a fairly simple demonstration, again, based on one of Tim Close's papers, and taking a fairly simple morphological phenotype that they analyzed and found some candidate genes. At the time, there was no genome for cowpea. And so one of the ideas behind this use case is, okay, now that we've been able to integrate some of their genetic data with the genome that they're in the process of publishing, using the tools that we're developing, how much easier has that basic process become? And one thing that I'd like you to just pay attention to in this use case is the fact that all of the orthologs in the other sequenced legume genomes, none of them will have the same names in the current genomes. It's only the Arabidopsis ortholog that they found that has kind of retained its identifiers, which is, it's kind of a trivial problem, but it drives people crazy when they're trying to, sort of leverage what they find in the literature with where the genomic resources have gotten to since the paper was published, and this was only a few years ago. All right, so we've been leveraging some software developed by model organism groups, primarily in the animal species. So this Intermine is actually a tool that was developed by the Flybase Consortium, but it's a very nice way of putting disparate types of data together and interconnecting a lot of different data sources. So we took the genetic data that we could get out of various spreadsheets, put it in, took the SNPs from the work that they had been doing, that at that point was a SNP chip, had the mapping to the genomic positions, and so it was very easy to see that the QTL, which they had associated with six or so markers, could be easily linked up to one of those markers that was the highest association with the phenotype. There are a number of mapping populations with genotypes in those populations associated with them. And if you scroll down, you can see, okay, the marker, Intermine can be sort of taught to do a lot of genomic computation work, fairly simple stuff, just like saying, okay, well, what's the nearest flanking gene to this marker? And now we've got ourselves a gene to work with. So now we've got a nearest gene to the marker that happened to be the closest on their SNP array. And then finally, we're calling out to a service now being provided by the legume information system that says, okay, well, I know about a lot of resources out there for cowpea. If I have a cowpea gene, what can I do with it? And what we're going to do is jump over to this tool called the Genome Context Viewer at the Legume Federation site. And that immediately is putting the cowpea genome, a little track from the cowpea genome. So this is, I forget which chromosome, but these are all genes that have been annotated according to the gene family assignments that we've put them through. And we're basically just pulling in new genomic segments for other legumes that we have present in our system, including non-legumes like Arabidopsis. And so what I really want to stress is that we haven't actually aligned any of the genomes directly to one another. All we've really done is to assign all of the genes from each of the genomes to a common set of gene families. These are actually from phytosome and defined by hidden Markov models that it's pretty trivial to just take the protein sequences, find the best match, and now you have what you can think about as a sequence of gene family assignments that makes it very easy to pull out syntenic segments like this. And we can see that there are some homologous segments that are further diverged from previous duplication in the legumes. You can see copy number, presence-absence variation here pretty easily. And you can see in the Arabidopsis genome, there's this triplication event with some fairly severe fractionation. But we do have the Arabidopsis orthologs. I mentioned its identity has remained unchanged. We can hop over, well, I guess this is the ortholog. It's in a nearby column. So now we've established the identity of all of the genes that now have different names with the cowpea new gene in the genome. And we can hop over into a tree that we've built at the Legume Information System and see kind of how all of the synthetically discovered genes map within the context of the tree. And this is a fairly well-defined gene family. You see a couple of different clusters here from that whole genome duplication event with some additional duplication within this clade, where apparently those things are going outside of the synthetic contexts. So, basically, and finally, we can then ask similarly about the Arabidopsis gene, what can I do with that, and get over to another implementation of Intermine that's been developed by the group at JCVI that gives all of the information that I would want from that ortholog, which actually tells me that it's involved in leaf development. So that's a fairly straightforward use case, kind of demonstrating the sorts of things that we're hoping to enable within, or actually are enabling at this point within the system. So just to kind of talk about what we see as being the challenges of being part of a dynamic, multifaceted, multi-organizational research community. There are a lot of overlapping resources that don't really have a lot of coordination between them outside of projects like ours, which is loosely coordinated at best. As everybody knows, genomic and genetic data are growing rapidly. It's, I've discovered just being at this conference that several of our genomes are quickly going to become obsolete for much better versions. But the value of most of the data that we're dealing with is really proportional to how interconnected it can be with other people's research. It's relatively easy to find genomes out there on FTP sites, but when people aren't really doing anything with them, they become much less valuable fairly quickly. And of course, we all know about cases of unfunded resources basically going extinct quite rapidly. It is hard to work with other people. You hear a lot of talk about making interoperable resources, but interoperability is just kind of a potential. It's not actual interoperation. And so, there's a lot of effort that goes into developing nice software, but without actually going that extra effort to work with people to try to make your things actually talk to theirs, you actually won't get any interoperation. It's very important to include specialists in each of the crops into your efforts, because there are obviously going to be unique biological properties to all of the different crops that we might want to deal with. And of course, we want to be able to use comparative genomics not only to kind of understand basic evolutionary biology, but also to really study agronomic traits and figure out how to leverage information in one species into another. So as I kind of alluded to, some of this is kind of technical, but a lot of it's really just social. So our project is really focusing on taking visualization tools and analytical tools and trying to get them into the hands of researchers, working together with other groups who are involved in the development of the software, and really, try to do everything from, report bugs that our users have discovered to helping them with documentation and things like that, which is, it's not very glamorous, but it's pretty important when you're trying to develop an ecosystem of research. We really want to focus on developing methods that really enable sharing among these disparate databases, and encouraging standards and protocols is really part of that. So as I mentioned, interoperability can only take you so far without actually making a strong effort to work with other folks. So a federation, has a long history in computer science of actually meaning something. So federated databases have been a technology that's been talked about for a long time. I would say that our project, we're sort of conceptually federated in the sense that we're thinking about our architecture in a way that we really want to allow the members to retain as much autonomy as they want. And we're really thinking about when we do develop new things, or we're choosing between various tools that we want to adopt, we want to think about really, well, what can really connect the sites without making them overly dependent on one another. Because, as I mentioned, we love our partners now, but we know that they won't all be with us in a few years. So we don't want to become necessarily too dependent on them. And there are some previous projects that were kind of in this space. Some of you with the GCP may remember a few of these. So these are the groups that we're working with. The funded partners within this are the LIS group, the JCVI group with the Medicago genome, Peanut Base, which is out of Iowa State, and Civers, which is a massive plant, it started out as a plant sciences infrastructural project with NSF, and it's since diversified to being less focused on plants and more inclusive of other sorts of scientific endeavors. And then we have a number of people that we, like the Kauke group that I mentioned, we're kind of reaching out to them and trying to figure out how the techniques that we're developing can be useful for them. Some of you may be familiar with some of these tools. As I mentioned, we're not building everything ourselves. When it makes sense, we're adopting technologies and encouraging other people to adopt the same technologies to the extent that it makes sense for them. And when it does make sense, we're also contributing our own software into the system. And in terms of federation, I'll just say, for a long time, my institute had kind of a conceptual problem with open source. We've put that far behind us, and I'm really delighted the way that some of the new resources out there for just kind of doing software development in an open way really encourage this sort of networking and social aspect of software development. So this is kind of a representation of the system with all the different players and the different tools that they've adopted. And I don't want you to really think too hard about this, but I did want to just kind of suggest that there's a sort of rough analogy between what we're trying to do and some themes in population genetics, where you do have a sense that there is information flow between these different things, but there's also value in isolation and kind of local adaptation of these individual resources to their users. But I do want to focus on this central data store, as we call it, which currently is being maintained at CIVERS. And what that is it's kind of our attempt to engage in what are known now as the FAIR data principles. I think this came out of a UK group who described it as having data be findable, accessible, interoperable, and reusable. And they place a very heavy emphasis on metadata standards and persistent identifiers. And in some ways, at this point in our development, this is really not much more than a fancy FTP site with some standards for how we're organizing data, but it does have metadata-based capabilities, and we're trying to encourage people as much as possible to sort of contribute the data that they're spending their time on curating into this commons area so that the systems that other partners are developing can kind of feed off of the same central resource and hopefully make the information more consistent between all of our sites. So one example of that is when we finally managed to get a number of our genomes kind of systematically put into the system. Eric Lyons at CIVERS has a system called Koji that was able to ingest the structures that we've been putting in there for the genomes and make what is called a private notebook, or well, it will be a public notebook for the Federation that contains a number of our genomes. And then all of the things that you can do with his system, like generate Synteny plots with KS distributions, are quickly available to people working with these genomes. And this will get you down to the level of individual sequence comparisons between your genomes if you want. Another example would be a tool that we developed for the legume information system, which is just a GIS viewer that allows you to see germplasm from the GRIN collection with trait data. This is photoperiod sensitivity for common bean accessions. And now we're able to put information about accessions that may be of interest to us. So for example, Medicago hap map accessions with traits from their GWAS experiments. We can now basically launch the GIS viewer with a reference to those specific trait values and see how they distribute over the map. So we're kind of integrating information about the geographic coordinates from the GRIN database for these accessions with the trait data that's now being represented in the data commons area for the HapMap data. And here's an example of population structure with the cowpea core collection that we've done in a similar way. So what you can do is, using the metadata-based capabilities of this, you can basically try to inject some intelligence in the system about what are the sorts of resources that the user's looking at and what are the tools that they can use to actually do something intelligent with them. So for example, I've got a VCF file here from our HapMap Medicago project, and it is able to understand, which reference was used to call the variants, and the fact that you could launch a genome browser to start looking at that. And the genome browser is pulling its gene annotations out of the data store. Those things have been annotated with specific links into other parts of our system so that you could then, for example, see, okay, well, this is where this Medicago gene falls within the context of the greater clades. Similarly, with a view like this that I showed you before, you could say, okay, well, here's an interesting little tandem duplication. What's that going to look like in terms of the variation? And you can see that very little reads are mapping there, presumably because it's hard to map them appropriately. Okay, so gene families have really been central to our development. We've been working with the phytosome families, both with respect to the legumes, and we've really been using the models that they built for angiosperms, and then included some outgroup information into our system for Arabidopsis and some of the other members of the nitrogen-fixing clade. And we've been inspired by many of the things that phytosome already does to kind of extend those. You know, basically, once they've defined the families with Markov models, it's quite easy for us to assign new genomes to them, use their definitions, and then build the new trees off of them. We are looking into the value of kind of building our own legume-focused trees. And this is kind of work in progress. What it's going to mean is that a lot of our tools that are really depending on these gene family assignments as being kind of the core are going to need to get revisited. But I think this notion of gene family assignments is really a strong navigational principle within the member sites. So you can do a lot of things by doing very little. So once we've assigned all these things to the gene families, here, for example, is peanut base. It's using the genome context viewer at LIS by just linking its gene out to it. And you can now see the two wild diploid progenitors lined up against one another. Our algorithms for doing the alignments kind of take into account things like inversions and whatnot. And so you get a very easy way of presenting the information about what the correspondences are between these genomes. And eventually, when we get the tetraploid in here, that will allow us to compare what those subgenomes look like against what the putative progenitor genomes are. Here's another example from the Medicago world. We've actually got a Medicago sativa accession, which sativa is a obligate outcrosser, so it's very heterozygous, and cultivated alfalfa is a tetraploid. We've been working with the diploid, and you can see that it's very easy to match up the two haplotypes against each other and see what looks like some interesting kind of gene content differences between the haplotypes. We're pulling this in a kind of a federated way so that not only are we getting that alfalfa genome, which is coming from one data provider, but also pulling in, say, Medicago HapMap data and getting all of the other genomes that have been assembled for the species and annotated. Pretty easy to visually assess content differences between them. This is an example from the chickpea genome, where we've got we included the Kabuli chickpea genes in our trees. We're able to link out to a resource that incorporated the wild chickpea, reticulatum. We get both the Desi and the Kabuli aligned together, as well as the wild species, and we reach out to one of our partner sites at the Cold Season Food Legume Database for the sequence information from that, and we can actually dynamically incorporate that into the tree. And this is showing us basically that the Kabuli is a little different. Actually lost an N-terminal domain for this gene, which is responsible for the difference in seed color between the two. We're doing some other things, developing kind of a modernized version of the old CMAP software, trying to allow you to take QTL experiments to find in different genomes, use markers on the genetic maps to link you onto the genome within the species, then use the macrosynteny between the species to cross between, say, common bean and soybean, and then get back into genetics for soybean. And again, these things link into the various other parts of our system using these kind of brokering services that I described. And one thing that I wanted to show off, this is kind of making the point that GWAS experiments have a lot of issues in terms of false positives because of population structure and those sorts of things. But if you're able to look at GWAS from kind of a multi-species perspective, you can take away a lot of those confounding factors. And so what we've done is adapted a tool called ZBrowse for looking at GWAS data. We've made it multi-species aware, so we've got Medicago GWAS data and Arabidopsis GWAS data. And this is now using services provided from the genome context viewer that I've been showing to kind of find the the regions that have similar gene family assigned content, and you can see we've got kind of a small peak. It's not all that significant, but we see it in Medicago, and we see that, in fact, the genes under the peak for Medicago are sort of also under the peak here, even though the region itself has gotten a little bit scrambled between the two. There's quite a bit of power there in being able to translate between the two species. Well, I see that I've quickly run out of time, so I guess I won't get to my more ambitious use case, which was to look at some R genes. But, well, maybe I'll just run through it very quickly if I can maybe have, unless anybody wants to ask questions instead. Okay, I'll just go ahead. So I took the example of the pigeon pea resistance genes for soybean rust. It's been mentioned a few times, presented by the keynote speaker. Took the sequences that were published as part of that paper, used our tool for doing annotations to our gene families as well as some other functional information, added them into the tree. They're clustering with the other pigeon pea genes that they belong to. You can quickly put them into context with some of the other genomes. So as suggested by the abstract from the paper, we've got common bean and cowpea, as well as soybean with just one copy there. We've got some interesting, probably assembly issues going on with the pigeon pea genome that you can see quite easily. We can also kind of take a different perspective and say, well, if I were to take all of the genes from this clade and just ask about the contexts that they reside in, regardless of whether they're syntenic, I can pull those into the viewer, kind of cluster them based on shared gene content, and then align within the groups. And so I see that the warm season legumes are all kind of grouping up here, and I've got chickpea and Medicago down here. And we can do these similar sorts of games, go over to resources that have different versions of the common bean genome that are less well assembled, pull together scaffolds that are sort of bridging that gap, and also see what's going on with the Medicago, a little bit of copy number variation, but there's certainly not as much as in the warm season legumes. And finally, we can kind of use these abilities of the system to create sets of genes based on phylogeny or based on synteny to populate Intermine with these lists. Intermine is really quite powerful in terms of dealing with lists, and you can do things like gene ontology enrichment or pull expression data for the multiple species into it and see kind of how the different genes from this cluster are behaving in terms of expression context. So that was, I guess, more information than I should have tried to put into a single presentation, but I'll leave it at that.